right, so in this chapter, we're going to be talking about basic genetics, which is like Mendel and Punnett squares and some of the simple types of crosses. So before we jump into the actual meat of genetics, these two formulas, the product rule and the addition rule, are on the formula sheet, so I wanted to address these. You probably are never going to look at the formula sheet, this section of it, and use these from the formula sheet, but it is likely that you'll use one of these, particularly the product rule, when you're solving some of the genetics problems. So I want to run through these real quick. So the first one, the product rule, says that the chance of any two independent events, both happening, can be calculated by multiplying their individual probabilities. So let me explain what that means. So for example, if we want to know the chance of rolling a 6 on 2 dice, so this is kind of the AND rule. So we know that the chance of rolling a 6 on the first die is going to be 1 out of 6, right? Because a die has 6 sides and only one of them has a 6 on it. And then what's the chance that the second die has a 6? It's also 1 out of 6. The product rule says we can multiply their individual chances to get the chance of both of them happening. So 1/6 times 1/6, I have a 1 in 36 chance that both dice would come up 6. Here's the second one. Suppose there's a 1/4 chance of rain today and a 1/2 chance of wind. What are the chances we will see rain and wind? So we take our individual chances, a 1/4 chance of rain, times a 1/2 chance of wind, and we get a 1/8 chance of seeing both rain and wind. So we're going to apply this to genetics a little bit later, but I just wanted to explain what the product rule says. All right, our second one, the addition rule, you're not as likely to use this one, but I will show you. So it says that the chance of one of several mutually exclusive events, things that can't happen at the same time, occurring is the sum of their individual probabilities. This is also called the OR rule. So what are the chances of either A or B happening? So you can sort of satisfy the criteria two ways. So what if we roll a die, we only roll it once, and we want to know what are the chances that it comes up either as a 1 or a 6. So you can't get a 1 and a 6 if you only roll the die once. But since one of the sides has a 1 and one of the sides has a 6, we have a 2 out of 6 or a 1 out of 3, if we reduce that, chance of meeting our criteria, because there's two ways we can meet our criteria. We can roll a 1 or we can roll a 6. Here's one more. If all students have a gray, a black, and a teal polo, and they randomly pick one every day to wear, what are the chances somebody is wearing gray or black today? Well, there's a 1 out of 3 chance they're wearing gray, because there's 3 polos to choose from. There's a 1 out of 3 chance they're wearing black. So there's a 2/3 chance that we meet that criteria of wearing either gray or black. Different from the first one, because in this scenario, we're not asking the chance of doing two things at once. In fact, in this scenario, it's not possible to be doing both things at once. But there's two ways to sort of satisfy the criteria. All right, so let's go ahead and jump into genetics. So genetics is based on probability. Just because we predict that 3/4 of the plants are going to, say, be tall, doesn't mean that if I have exactly 4 plants, 3 are going to be tall. But we do know the bigger our sample size, probabilities do work. We will get close enough if we solve with, say, a chi-square test, where you do observed and expected and you see if you're close enough, it will pass a statistical test saying that, yes, it's close enough that it falls into those probabilities. Okay, so Mendel is considered, I know everybody's heard of him, he's considered the father of genetics. He was a monk who worked with garden peas, and he was very interested in crossing the different kinds of traits to see what he got. Okay, so it turns out that garden peas were a really, really good choice because of several reasons. The first one is, and this is kind of a unique thing for plants in general, they were self-pollinating. They still reproduce sexually, but a single pea plant, you can see this in the diagram here, has both male and female parts. So if he just left this plant alone, it could fertilize itself. And he would therefore ensure the paternity of the cross. He didn't have to guess, if there could be contamination, because the plants would simply fertilize themselves. And that's called self-pollinating. These other reasons are also important. The traits were simple and easy to see. Every trait he studied pretty much just had 2 versions, and they were very obvious. And the third thing, they grow fast, easily, and have a lot of offspring. So every pea is an offspring. So one pea plant could produce 100 offspring, and then a couple months later you have a whole nother generation and a whole nother generation, and they were very easy to take care of. This is actually all the reasons why humans are very difficult to study. Our traits are not all simple and easy to see. Some of our traits are very complicated. Like if you think about our skin tone, there's not just two. There's a whole range of eye colors, skin tones, hair colors, things like that. So a lot of our traits are complicated. We don't grow fast. We don't grow easily. We don't have a lot of offspring. So things that would be good for studying would be things like mice, rabbits, fruit flies. Think of things that reproduce sexually. Bacteria would be a bad choice because they reproduce asexually. But something that reproduces sexually, has lots of babies, and has simple traits that we could see. Okay, so what he specifically did was he took purebred plants that he knew, because remember they self-pollinated, so he kind of knew generation after generation this plant always made tall offspring. And in this other area of the garden, this other plant always made short offspring. And then he would crisscross them by pollinating one, taking the pollen from one and putting it on the other. He called the original purebred parents the pea generation. P for parental. But you can also think of them as being purebred because he always started with purebreds. The offspring of these parents would be called the F1. F actually stands for filial, which means son. So it's the children, basically, of crossing the two peas. And then the grandchildren. He crossed two of these with each other, and this was really important because he wouldn't have realized what he realized if he had not gone that second step. So these were called the F2. In theory, yes, you could go F3, F4, or whatever, but that's not necessary. Really, what he did was perfect. Parents to their offspring, and then cross the offspring. Okay, so here's an example of what he did. So we have tall and short. Now what he found is every time he started with a pea generation of pure parents, when he crossed the two versions of the trait, one of them disappeared in the F1 generation. So in this scenario, as you can see, all the offspring came out tall. This happened every single time. Every trait he tested, one trait sort of covered up the other. And this is going to lead us to his first principle, the principle of dominance, which says some traits cover up or mask other traits. He also figured out that his peas had two copies of sort of code for the traits. He did not know genes. He didn't know meiosis. He didn't know chromosomes, none of that. But he figured out that they had to have two factors, which we now would know that they're two copies of the gene. They're diploid, right? So he would use the first letter of the dominant trait. That's kind of the way we usually do it. Use one letter. And the original tall one would have been big T, because it's purebred for tall. And the short one would be little t, because it's purebred for short. And when he crossed these, he figured out, again, didn't know about meiosis, that each parent, even though they have two copies, is only giving one of those two. So this parent could only give a big T, this parent could only give a little t, and all these offspring, even though they were tall, were actually big T, little t. Then he crossed two of these. That's what crossing the F1 means. So he crossed big T, little t with another one just like it. Even though these look identical to these parents, they're not actually identical, right? Interestingly, he noticed in the second generation, the short plants came back. And that showed that short wasn't just like wiped out, it was just sort of masked. Why did it come back? Well, because this time, unlike in their original cross where this could only give a big T, each parent now had two options. It could give a big T or it could give a little t. So we had multiple things we could get. We could get big T with big T. We could get big T from the first parent with little t from the second. We could get little t from the first parent and big T from the second. Or we could get little t from the first parent and little t from the second. Now, quick comment, we don't ever write little t, big T. We would say that these are the same. But notice there's two ways to get big T, little t. And that's kind of where the addition rule could come in. And so anyway, what he discovered was he always got a ratio of about 3:1, or 3/4 showing the dominant trait, 1/4 recessive. And notice these numbers, these are not exactly 3:1. But because he tested large numbers of P's, it definitely would pass a chi-square test. Statistically, it's 3 to 1. Okay, so he came up with 3 principles from this. The first principle was the principle of dominance. This is easy. It says some characteristics or traits mask others. He called the ones that mask the others dominant. It doesn't mean that big T somehow deactivates little t. That's really important. That's like a misconception. What was probably really happening in this case is big T codes for making a lot of hormones that make the plant grow tall, and little t is probably a version that codes for making very little. So if you have at least 1 copy of big T, you would have enough of the hormone to grow tall. Whereas if you had little t, you would be short. But the big T doesn't actually do something to the little t, just to clarify. Anyway, we call these versions of the gene alleles. Make sure you understand that. It was 1 gene, in this case, which controlled height, but there were 2 versions or alleles. This is where homologous chromosomes come into play. Homologous chromosomes, remember, were not identical. They come originally from, one came from your mom, one came from your dad. So a pea plant could have 2 copies that were identical, but it could also have 2 copies that were different because each chromosome came from a different parent originally. These are just some of the traits he tested: round versus wrinkled, yellow versus green. And again, he got very lucky because every single one of these traits showed the principle of dominance. Like, he never crossed purple and white and got lavender. That would have been very confusing. If a plant does that, which some species do, that's not showing the principle of dominance. That's a non-Mendelian trait that we'll talk about next week. But right now, we're talking about what Mendel did, and all of his traits followed these rules. The second rule is the principle of segregation. The principle of segregation just says, again, he didn't know they were traits or genes or meiosis, but we now know every individual has 2 copies of a gene, but during meiosis, they only give 1. And that's what segregate means. It just means to separate. So the principle of segregation simply says that even though you have 2 copies of every gene, you randomly only give 1 to a sperm or an egg. That's all segregation says. The third principle is the principle of independent assortment. Independent assortment says, and this is builds on the segregation principle, that every pair segregates randomly compared to every other pair. In other words, every time meiosis happens, it's random which copy the gamete receives. Now, this will make more sense when we get into dihybrid crosses, but this is where the 2 to the n came in. So, for example, if an organism is big B, big C, little c, for 2 different traits, what we're saying is that n, in this case, is going to be the number of different letters. So we have B and we have C. So n is going to be 2. The haploid number of this organism would be 2. So there are 2 squared, or 4 possible outcomes when meiosis happens. It could give big B with big C. It's going to randomly give a copy of B and a copy of C. Or it could give big B with little c, or little b with big C, or little b with little c. Those are all the possible outcomes when they randomly, through meiosis, give 1 copy of this chromosome and 1 copy of this chromosome. So this one would actually be 2 to the 3rd, or 8 possibilities. And you could write them all out. They could give big B with big B, C with big D, big B with little c with big D. It's all the possible genes that could be given to a sperm or an egg in meiosis. That's what they're talking about here. We had this picture up last chapter too. So this is what this is showing. This is showing meiosis 1 and meiosis 2. We're not going to talk about the two stages, but if they line up this way, this one's going to give capital A and capital B. Whereas if they line up this way, it can give capital A with little b. So it's metaphase of meiosis 1 where it happens, but that's what he's talking about. Other genetic vocab we're going to wrap up with: a gene is a DNA code for a trait. We usually use the dominant version. Purebred, true breeding, or homozygous mean the 2 letters are the same. Homozygous dominant would be capitals. Homozygous recessive would be little. Heterozygous, the letters are different, big, little. Genotype is the genes. Phenotype, the physical appearance. And we're going to talk about this later, but phenotype can be affected by the environment.